So the liver happens to be the largest internal organ that we have. Remember when we were talking about the integumentary system, we said that skin collected together is the largest organ, but it's not inside of the body. So liver happens to be the largest internal organ. It's found in the abdominal cavity, right underneath the diaphragm, on the upper right quadrant. It is very reddish brown in color because of all of the blood supply that it has, and we'll talk about how it has a dual blood supply. The liver, because it's so big and heavy, and it's, again, right underneath that diaphragm in the upper right quadrant, it has to be attached to the diaphragm and to the abdominal wall with different ligaments in order to carry the weight of the liver. So you can find what is known as the coronary ligament. Okay, and the coronary ligament kind of attaches it to the abdominal wall and to the diaphragm. And attached to that, or kind of an offshoot of the coronary ligament is the falciform ligament. Okay, and the round ligament. All of these are just different ligaments to keep the liver in place. The liver is made out of 4 lobes. The falciform ligament divides it into a big right lobe and a smaller left lobe. Those are your 2 main lobes of the liver. When you flip the liver upside down, so you're now looking at the inferior surface of the liver, you can find 2 other lobes. The quadrate lobe, because it has 4 sides, so again known as the quadrate lobe, and that is towards the gallbladder. And another one known as the caudate lobe. Caudate means tail. So the caudate lobe, that is right by the inferior vena cava. And in order to understand the blood supply of the liver, and I kind of got this, from another class, and because it's really important to understand the rich blood supply that the liver is getting. It's very unique in that it gets oxygen-rich blood, just like any other organ, through the hepatic artery. The hepatic artery is a branch off of the aorta, and again, it brings in oxygen-rich blood to the liver. It's not a direct branch off of the aorta, but, eventually the blood supply or the blood coming from the hepatic artery eventually does come from the aorta. It has another blood supply which happens to be deoxygenated blood. So this blood coming in is not to bring in oxygen to the liver. It is actually coming from the gastrointestinal tract, so from the small intestine and from the spleen and so on. It is bringing nutrient-rich blood, meaning all of the things that we've absorbed, all the goodies that we've absorbed through the small intestine would reach the liver through the hepatic portal vein. So again, 2 blood supplies, hepatic artery that is rich in oxygen, and then the hepatic portal vein brings in blood that is deoxygenated. So again, the aim of this blood supply is not oxygen, but the aim is to bring in all the nutrients that were absorbed from the small intestine, with the exception of fat. So fat is a different thing. All of that blood, all of these 2 blood vessels are going to give out different branches. The blood is going to mix together, and the liver cells, known as the hepatocytes, are going to use that blood in different things in order to perform the function of the liver. And then all of that blood is going to exit the liver through the hepatic vein, which is oxygen-poor blood. The hepatic vein eventually will reach the right atrium, just like the rest of the body, through the inferior vena cava, and so on. Okay, so again, 2 blood supplies going towards the liver, hepatic artery rich in oxygen, hepatic portal vein brings in blood that is nutrient rich. All of that blood mixes together in these different lakes known as sinusoids that are found in between the hepatocytes. This helps the liver perform its function. And then all of that blood is going to be drained out of the liver and eventually go through the inferior vena cava to reach the right atrium through the hepatic vein. So let's take a look at how these blood vessels will break down. So again, you can see here a branch off of the hepatic artery and a branch of the hepatic portal vein. Remember, this is bringing in oxygen and this is bringing in nutrients that we've just absorbed from the small intestine. And all of that blood is going to mix together in what are known as these little lakes or hepatic sinusoids. This is where these liver cells or hepatocytes are going to take their oxygen and take those nutrients, whether they're amino acids or glucose or whatever nutrients that we just absorbed, and utilize them to perform different functions. Remember, we talked about the liver kind of being the kitchen of the body. Well, this is how it gets the ingredients to cook up the different things our body needs. Another thing the hepatocytes do would be to produce bile. So you can see it producing all of that green bile, and that green bile is going to drain into these bile ducts. Now, going back to these sinusoids, all of that blood now that is, at this point, all of it is now deoxygenated, will drain towards that central vein, and all of these central veins will gather together to become eventually that hepatic vein that again drains the blood into the inferior vena cava. So this is how these hepatic lobules are organized. That's how, if you take a microscopic picture of the liver, this is what it looks like. You have these hepatic lobules, you've got that central vein draining all of the blood, and you'd find at the edges of these lobules, you'll find a branch of the hepatic artery and a branch of the hepatic portal vein. Here again, you can see, in a bigger picture, you can see how that branch of hepatic portal vein bringing up the nutrients, the ingredients, that branch of the hepatic artery bringing in the oxygen, all of that is going to combine and mix together in the hepatic sinusoids. You see here that all of these, what are known as the hepatocytes, these are all of the active liver cells that are performing their function. They're using the oxygen, using those ingredients to cook up different things, whether they're plasma proteins or glycogen or whatever the body needs at the time. And these hepatic sinusoids also have Kupffer cells. Kupffer cells are cells that were named after the scientist named Kupffer. These are macrophages that have the ability to phagocytose anything that might be coming in from all of that blood that is pooling into these sinusoids. Okay, again, all of that blood, after it's being used, will drain into the central vein. Another thing, again, to keep in mind is that these hepatocytes secrete bile, and all of that bile will gather into these bile ducts. Now, these are the major functions of the liver. The liver basically can take any nutrient, okay, whether it's a protein, amino acid, or a glucose or fat, and can change it into different things. So let's talk first about what the liver can do in regards to carbohydrate metabolism. Well, if we have too much glucose, the liver will store it as glycogen. Okay, but on the other hand, if we need glucose, it can break that glycogen into glucose and release it into the bloodstream. It can even convert non-carbohydrates into glucose. So it can take amino acids and change them into glucose. It could take fatty acids and change that into glucose. And that process is known as gluconeogenesis, because you are generating new glucose out of pretty much nothing, out of a non-carbohydrate. Again, a process known as gluconeogenesis. The process of converting glucose to glycogen is known as glycogenesis. And the breakdown of glycogen into glucose is known as glycogenolysis. The liver function in regards to lipid metabolism, it can oxidize these fatty acids. It can use fatty acids and attach it to a protein to make lipoproteins. It could take lipids and attach them to phosphates to make phospholipids. It can also make cholesterol out of them. Remember, then cholesterol can become different kinds of hormones. That cholesterol will go to the different glands, and the glands will use them to make cholesterol or steroid hormones. It can even convert, like I said, these fat molecules into glucose or carbohydrate molecules into fat. So again, liver has the ability, these hepatocytes have the ability to pretty much mix and match everything and convert things into different things. In regards to protein metabolism, it can deaminate amino acids. So what really happens is that a lot of these amino acids are pretty much recycled in the body. The body is a very conservative thing, and it doesn't want to get rid of these amino acids, even after it breaks down proteins. But at a point, these amino acids tend to become very old, and they would have to eventually break them down. The liver deaminates it, which means to remove that amino group, NH3 group. It'll eventually form urea, which is a waste product, okay, that would have to be secreted or excreted out of the body. Remember, the liver is responsible for making plasma proteins, which are eventually responsible for the osmotic pressure of blood. And as we already mentioned, it can convert amino acids into glucose, but it also has the ability to convert amino acids into other kinds of amino acids, which is pretty awesome when you think how the liver can just play around with these different molecules depending on the body need. The liver is a storage area for glycogen. So remember, after a meal, if we eat up too much glucose, we'll store up some glucose as glycogen in the liver, other in the skeletal muscles, and then if we still have more glucose in our bloodstream, it'll start storing it as fat. Okay, the liver is also storage area for vitamins A, D, and B12. It also stores iron, and a little bit of blood is stored there. Not literally, blood always has to be circulating, but because of the rich blood supply, it is considered kind of like an area where blood is technically stored. Blood filtering, the liver will filter blood, especially due to the action of these Kupffer cells, which are macrophages or phagocytic cells that are going to remove damaged red blood cells. They'll remove any foreign particle that try to come in with the nutrients. It can also detoxify, so it removes toxins from blood, and it secretes bile. Okay, so you want to kind of think of these functions in somebody that might have maybe liver failure. So in liver failure, some of the things that, these patients would experience would be the inability to make plasma proteins, for example, okay? And that would lead to decreased oncotic pressure, which means that they're going to swell up in the different parts of the body in the form of edema. So edema, especially in the lower limbs, because that's where gravity will take that water, or surrounding the other organs, like in the pericardial sac or in the pleural cavity and so on. All of that would be due to the low plasma protein levels in the bloodstream. Now, liver replacement or liver donation. That liver is really required to maintain life. So you've seen all of these different functions of the liver, and without these functions, it is really, we're pretty much incompatible with life if our liver is not functioning at a minimal level. If we do have at least a quarter of a healthy liver, that is enough, because the liver is capable of regenerating itself as long as the liver tissue is healthy. And that's why, again, donating parts of the liver, okay, is a thing, because, again, the liver can regenerate. Due to all of the functions of the liver, though, if cancer tends to spread to the liver, damaging most of it, again, that is incompatible with life. It is considered a late stage kind of cancer. And again, in order to regenerate, you need at least a quarter of it to be healthy to be able to keep up with all of the needs of the body. Now, hepatitis is the inflammation of the liver, and the most common kind of hepatitis are usually viral hepatitis. Sometimes they can be acute or chronic. So acute basically means that the patient gets sick, and they get better and they're fine. Okay, so there are no signs or symptoms after the attack. Chronic conditions are a little bit different, and that's where, they get sick, and then the virus tends to remain dormant in the liver, so they can become carriers and they can infect other people. And then they could get these flare-ups where they start experiencing symptoms and so on. And that chronicity tends to happen more with hepatitis C patients. Hepatitis C, again, has the ability to become chronic conditions, and it is also precancerous condition if it remains untreated. Some people actually get viral hepatitis without any signs and symptoms, but they can become carriers. Some of the hepatitis viruses are bloodborne, other are through food. Okay, though it can be transmitted through food or body fluids or objects that are contaminated with feces that contain the virus. So the ones that are transmitted by food, the person that has the virus is shedding the virus in their feces that can eventually contaminate food. Antibiotics are not effective against viral hepatitis. Remember, viruses are not bacteria. Antibiotics can kill bacteria, but they do not kill viruses. Now that we know that hepatocytes make bile, okay, what is the composition of bile? So we one thing that you want to keep in mind is that we tend to always couple bile with the gallbladder. Okay, the gallbladder does not make bile. So even a person that has removed their gallbladder, they are still making bile. The gallbladder is there to concentrate and store bile. Okay, but again, hepatocytes or the liver cells are the cells that produce and secrete bile. You can see that there's a continuous secretion of bile by hepatocytes. And then it will be transmitted to the gallbladder in order to store it and then release it upon eating, especially if they're fatty diets. So what is the composition of bile? Well, there's lots of water in there. There's lots of bile salts, and those bile salts are produced from cholesterol. So the hepatocytes will take cholesterol and change it into these bile salts. Bile salts are really important because they emulsify fat. And if you're not familiar with the term emulsification, it basically means to break down fat into smaller cells to help the lipases in the small intestine to digest fat. So fat tends to gather in bigger molecules. So think about, when you watch these commercials where you have this fat stain or oil stain in a sink and they put a drop of detergent and it just pretty much, disappears, okay? That process is known as saponification or emulsification. So the bile salts that are found in bile produce that same effect that soap does. And again, the term saponification comes from that function. Where, again, that bigger molecule of lipids is going to be will be broken down into smaller parts so that the lipases can work and digest fat. So bile salts are really the only component of bile that has any digestive function. Everything else has no digestive function. Bile pigments are also a composition of bile. These bile pigments, namely, are bilirubin and biliverdin, and those are derived from hemoglobin breakdown. So if you guys remember back when we were talking about what happens to an RBC when it gets old, we talked about the breakdown of hemoglobin into heme and globin. And we said globin is a protein, but the heme will change into bilirubin and biliverdin. It gives the bile its greenish discoloration, and then when these two pigments reach the small intestine and the large intestine, they change through bacteria into stercobilin and stercobilinogen and so on, and that gives the feces their very characteristic brownish color. Cholesterol and electrolytes are also composition of bile. Now for the gallbladder, and that is the sac, okay, that is found on the back of the liver. So this again is an anterior view. This is a kind of a posterior view. And now you can see the gallbladder. Again, it stores and concentrates bile. It does not make it. When the gallbladder contracts, which is mostly due to the effect of the hormone CCK or cholecystokinin. The bile will pass through that cystic duct. Okay, and I'll go through the duct system in a minute, but all of that bile again will be released into the duodenum. Remember, it will join the pancreatic duct, and both of them are controlled by the hepatopancreatic sphincter. So I want to go back more than a couple of slides, actually, just to go over the duct system. So there we go. So you can see here, the liver would be right up here. You've got your right lobe on this side and your left lobe on that side. And bile will be drained from each lobe through these two ducts. So you'll have a right hepatic duct and a left hepatic duct. They combine together to make a common hepatic duct. Now, the duct coming out of the gallbladder is known as the cystic duct. The cystic duct combines with the common hepatic duct to make this longer duct known as either the common bile or just the bile duct. And it's that bile duct that joins the major pancreatic duct to open up at the hepatopancreatic sphincter. Okay, so let's go back to where we were. And we'll talk about gallstones, which is a very common pathology. Well, we talked about how the gallbladder has to concentrate bile, and sometimes that, the gallbladder takes it a little bit to the extreme where too much concentration happens, and that'll start to precipitate different things like cholesterol or bile salts or these bile pigments. And that is how these little gallstones are going to be formed. Okay, so it starts off with these little tiny crystals that enlarge to produce gallstones. And again, the causes are either too much bile concentration or that the liver secreted too much cholesterol into bile, or it could happen due to the inflammation of the gallbladder, a pathology known as cholecystitis. How do we regulate the release of bile? That is something that we already know, where chyme is emptied from the stomach into the duodenum. That stimulates the duodenum, again, stimulates those stretch receptors, and also due to the presence of protein and fat, that is going to release the hormone CCK or cholecystokinin from the small intestinal cells. And CCK is going to lead to the contraction of the smooth muscles of the gallbladder and relaxation of the hepatopancreatic sphincter. It also brings in, or also leads to the contraction of the smooth muscles in the pancreas to release pancreatic juice. Okay, so all of that, CCK, will eventually lead to the release of bile and pancreatic juice into the duodenum. And the name CCK should tell you what it does. Okay, so cholecystokinin, if you break that down, kinin means to activate, coming from kinetic energy. Cyst and chole means the gallbladder. Okay, so to activate the gallbladder. What are the functions of bile salts? And we kind of already talked a little bit about it, but a couple more things to add here is that bile salts, again, is the only digestive part of bile. It helps through emulsification. Remember, we said that that means to break down these fat globules into smaller droplets, kind of like what soap does. Again, a process known as saponification. It doesn't really digest fat, but it helps in fat digestion because now that the bigger fat globules are now smaller droplets, that gives the enzymes, the lipases, it gives it more access to the triglycerides to digest them. And bile salts helps in the absorption of fatty acids and cholesterol by forming micelles. And micelles are little fat, kind of little balls of fat that we cannot absorb because of how hydrophobic they are. So bile salts surrounds it in order to make it water soluble and help us absorb them. Bile salts will help, because it helps in the absorption of fat, it helps us absorb these fat-soluble vitamins, which are vitamins A, D, E, and K. And bile salts are mostly recycled. Okay, so when it goes down through the small intestine reabsorbs about 95% of it back into the and eventually find its way back to the liver. We only lose about 5% in feces. And that is the end of the liver.